I'm Nicole Murdoch.
And I'm Kenzie Mohammed, and we explored tissue scaffolds for our iWonder project. So our question is, in the case of a cranial injury where we need a tissue transplant into the injury for the skull, what kind of biomaterial is most biocompatible for the scaffold that we're going to use? Our claim is that we must use a mixture of synthetic and natural polymers of bioceramics.
So when we started out the project, our topic was really broad. We started just researching anything that had to do with tissue engineering because we found the topic really interesting because both of us want to go in the medical field and it seemed really interesting to both of us. However, after doing some more research on the important components needed for specifically the scaffolding for tissue engineering, we found that one of the most important factors that we also found really interesting was the biocompatibility of biomaterials that are used. And then also after doing some more research, we narrowed down the project once more to specifically the correct biomaterials used to ensure biocompatibility within cranial injuries.
Recently, medical researchers have been researching 3D printing and tissue engineering as a way to replace tissue donors and organ donors for the human body.
So when tissues are 3D printed, you need to have a scaffold, which is then they're placed to hold the shape of the tissues when it's being 3D printed around the scaffold. Once these tissues are placed inside the body, the scaffold are eventually biodegrade and just leave the body. However, in order to have a scaffold to function with the body, we need to make sure that it's biocompatible, and this means that it's able to function within the human body and make sure it does not cause either an immunological or toxic response to these materials. And some of the most common biomaterials that we see to make sure that they're biocompatible are hydrogels, composites, synthetic and non-synthetic polymers, metals,
and bioceramics. Most important one for our project.
So our first biomaterial we're going to talk about is metals, which are very important because they have really high mechanical properties due to their ability to hold large amounts of strain, large amounts of strength, and they also have high ductility. However, there's also some downsides to having metals as a biomaterial. These include having reduced cell-cell adhesion. Sometimes biological materials within the human body are able to corrode the metals, and also they sometimes release toxic metallic ions, which can be very harmful to the human body. Usually, these materials are either made out of stainless steel, cobalt, or titanium alloys.
The second kind of biomaterial are composites. And composites are basically a mixture of different biomaterials, and it can be two or more. So for example, a good example is sometimes metals are a biomaterial that are used and then to reduce toxic shock and to reduce and to make the metal more acceptable in the body, they coat the metal with a natural polymer so that the body more easily accepts it. So that's what composites are used for. They're really useful, obviously, but the downside of them is that they must always be synthesized to mix all of these biomaterials together, so they can get really expensive.
So the next biomaterial I'm going to talk about is hydrogels, which are really important because they're hydrophilic, which allows them to absorb large amounts of water and reduces swelling within the scaffolds of 3D printed tissues. Hydrogels are usually created out of amide, amino, hydroxyl, and carboxyl groups. And also they're also important because they're easily modified on the surface by polymers. So overall, while hydrogels often have reduced biocompatibility, they're also really useful because you can easily modify their biodegradability or their mechanical properties to customize it for whatever tissue you need it for.
Next are bioceramics, and bioceramics are good. Usually they're used for bones and dental issues. They're very dynamic in their uses. You can apply them using load-bearing pieces or powders, or inject them so that they're more porous. So they're very dynamic in their uses and the way that we can apply them surgically. And then another thing is that they're usually made out of carbon phosphates, which are very close in chemical structure to normal bones, so that reduces the chance of toxic shock. And then also it makes them more osteoconductive, which means that they're more able to talk with cells around them, and cell signaling is more efficient that way.
Next, we have natural polymers, which are found in nature with very minimal artificial interference. They're very difficult to synthesize because they're really complex structures, which also makes them very expensive to make. They're also beneficial because they have a reduced chance of toxic shock when placed inside the human body. Some of these include cellulose or starch.
Next are synthetic polymers, which are the opposite to natural polymers. And what synthetic polymers are is they're polymers that are synthesized completely in the lab. They're completely artificial, or mostly artificial. And they're really good. They're really nice because they're predictable, because they've been made in the lab, and they're also easily customizable. But the problem with them is that because they're artificially made, they don't have that complex structure that natural polymers have organically. So that means that it's harder for synthetic polymers to be biocompatible and completely biodegradable, and that can unfortunately cause toxic shock and cause an immune response because the polymer is not biocompatible.
So to sum up the difference between the natural and synthetic polymers, both natural and synthetic scaffolds have many advantages and disadvantages. To name a few of these is that synthetic polymers are often a lot less expensive than natural polymers because they're less complex and easier to create. Also, synthetic polymers do not have the chance of bringing diseases with them. However, on the other hand, natural polymers have more efficient
degradability.
I was going to say degradation. And cell communication. Also, the last piece of evidence we have is in the skull. So the skull is made out of many different pieces. It's also made out of both bone and cartilage, which makes it really important to have a biomaterial that can both be used for hard and soft tissues.
That's dynamic, like bioceramics. Keep that in mind.
So also this skull can be really hard to engineer because the bone is flat instead of tubular like a lot of other bones are. And also, specifically, they found within the skull, excessive intrusion into the regenerative site can often cause or interrupt like bone regeneration,
which is not good.
Obviously.
So now that we have all of our evidence, we can conclude that the best kind of biomaterial for this specific question for an injury for a cranial injury is a mix of synthetic and natural polymers of bioceramics. And we've talked before about toxic shock. The factors that affected our decision in biocompatibility is the probability of toxic shock and how many times we'd have to re-perform the invasive surgery if something doesn't go well.
So we decided to use both synthetic and non-synthetic polymers for the scaffold for a few different reasons. First off, synthetic materials can cause toxic shock when placed inside the human body. So by doing a mix between synthetic and non-synthetic, we lower this chance. Also, synthetic polymers are more easily able to be created and customized. However, on the other hand, Non-synthetic or natural polymers are much more difficult to create, and because they're super complex, and because of this they're really expensive. So by having a mix between the two, we're both lowering the cost and lowering the chance of toxic shock.
So the second part of our claim is that we chose bioceramics, and the reason we chose that is because in the skull there are soft tissues and hard tissues because there are cartilage and bones. So that means that bioceramics would be most biocompatible because we can apply them as both soft tissues and hard tissue. And more importantly, the skull is bone, obviously, so bioceramics would be most osteoconductive because they're made out of calcium phosphates. So that's also a second really good thing that will reduce the chance of toxic shock.
So to rule out our other materials, first, metals, we found that those would definitely be way too hard for the cartilage portion. They also can cause toxic shock, so they can possibly be dangerous.
And then we eliminated hydrogels because we thought they would definitely be too soft and they wouldn't provide the protection needed for the skull at all, because our brain is in there and we need protection. We need something solid, which hydrogels can't provide.
And then lastly, we have composites, which, because they're able to be used for both soft and hard tissue, they could have worked, but because you have to like mix several different synthesize many different biomaterials together, it makes them much more expensive, and we did not have the need to create this super complex, expensive scaffold when we had the ability to use
bioceramics. Yay. So that was our iWonder project.
Thank you for listening.
Mohammed. And okay. So recently, medical researchers have been looking for a way to... Okay. Ready?
Ready?
We got
that. Gels. Because like they're able to surface modified, right, by like thingies.
Okay, it's fine. She's not fine, but it's fine.